Hello, everyone, and welcome to Epilepsy Research UK's Coffee with Cronutt the Sea Lion webinar. That's right, a session about a sea lion who's being treated with frontline treatments for epilepsy. My name is Professor Stephanie Schorge, and I'm a trustee of Epilepsy Research UK and also the head of the Department of Neuroscience, Physiology and Pharmacology here at University College of London. I am absolutely delighted today to be joined by Professor Scott Baraban from the University of California, San Francisco. Professor Baraban is the William K. Bowes Jr. Endowed Chair in Neuroscience Research, and his lab has led the pioneering research in interneuron therapy for the treatment of drug-resistant epilepsy, as well as a really unique angle on zebrafish epilepsy research. I first met Scott about 2 years ago at Epilepsy Research UK's 2019 International Expert Workshop on Neurodevelopmental Disorders and Epilepsy, where he gave a presentation giving the very first glimpse into what might become the Cronutt story. And I have to say the audience was completely hooked on his presentation on the potential of interneuron therapy for epilepsy. Today, Scott is going to be talking about his research and how a sea lion named Cronutt, who has unfortunately got epilepsy, has been the unlikely but extremely lovable beneficiary of Scott's groundbreaking work. So welcome, Scott. I'm going to hand over to you now.
Thank you so much for that wonderful introduction, and happy to be here this morning for me, this afternoon for y'all, to discuss some of our research related to interneuron transplantation and specifically what we've been doing with Cronutt for about 8 or 9 months now. So without getting too far, let me jump right in. I can't, there we go. So the entire premise of the research we've been doing here for almost 2 decades is based on a very simple kind of idea. Our brain has 2 types of neurons. The majority of those are excitatory neurons. They make up about 80% of all the cells in our brain. And the other 20% are what are called inhibitory interneurons. They're inhibitory because they express a neurotransmitter called GABA. GABA, when it's released onto other cells, hyperpolarizes them, or very simply brings them further away from firing. And so it basically dampens the electrical excitability of the nervous system. Interneurons play many roles in both normal and abnormal function. They're a heterogeneous group of cells, as you can see in this diagram to the left, which tries to simplify it. The green cell is an excitatory cell, and all the other types of cells are the different types of inhibitory interneurons that make contact with this cell. And so they are diverse, they're heterogeneous, and interestingly, when they don't work well, or they're damaged, or they're actually lost, as in cases of temporal lobe epilepsy, you have less inhibition. And as you can imagine, in a disorder like epilepsy, where there's excess excitation creating a seizure event, when you have less inhibition, the brain is more likely to generate these seizure-like events. So it's not surprising that modifying inhibition is a long time kind of goal of epilepsy treatments. And the way we've gotten there over the years is to use drugs. Those drugs are things like benzodiazepines that you're aware of, such as diazepam, that basically enhance GABA-mediated synaptic transmission. So they increase inhibition. But drugs, of course, work globally, and they're going to increase inhibition everywhere in the brain. And they're not very specific to the regions where you're specifically generating seizure events. And so we reasoned years ago that if you can put in new inhibitory cells that became interneurons and integrated in our brain circuits, that these cells would function to enhance endogenous levels of inhibition and could be therapeutic in epilepsy. To get there, of course, is not that straightforward. First, we have to know, where do these interneurons come from? Luckily, one of our longtime collaborators here, John Rubinstein, is one of the first labs to discover where in the brain, in the embryonic brain, interneurons are born. And it's a region called the medial ganglionic eminence, which you can see here multi-labeled in the middle. There are ganglionic eminences on both sides, and they produce the majority of interneurons of those 20% of cells that we have in our brain. But there are different flavors. So cells from the medial ganglionic eminence produce a subset of interneurons called parvalbumin or somatostatin interneurons. These directly impinge on excitatory cells, and they play a critical role in controlling excitability. other interneurons of the heterogeneous group are born in the other ganglionic eminences. A second longtime collaborator of ours years ago discovered that if you harvest or take out this little section of the embryo from a rodent model and you transplant it into another rodent, then those cells will migrate extensively in the host brain. And that's when I met John Rubenstein and Arturo Alvarez-Bulla about 2 decades ago, and first came up with this idea that maybe, just maybe, we could put new inhibitory interneurons into an epileptic brain. So summarizing now decades of work and jumping right into where we are at the moment, the bottom line is if you transplant these cells, they will integrate in the host brain. You can see that with these little green dots, little green cells on the blue background, which is the host brain. And you can see they distribute widely into this region. This is the hippocampus. Many of you know hippocampus is intimately involved in temporal lobe epilepsy. And interestingly, the hippocampus of patients with TLE have reduced numbers of inhibitory interneurons. And so we found over many years that these cells will integrate, they'll migrate, they'll become the types of interneurons we want. And most importantly, they'll functionally integrate in the host circuit. That means we can increase inhibition with these new cells. And so this gives us a great opportunity to test whether transplantation therapy, which is a one-time procedure, might be therapeutic in disease cases like epilepsy, where you have reduced inhibition. So what we've done over the years is test this idea using experimental animal models of epilepsy. We've looked at genetic models of epilepsy, which occur based on a rare gene mutation, a single gene mutation that causes the animals, like people with the same mutation, to have spontaneous seizures. And we've done this in acquired form of epilepsies, basically taking an adult animal model where you induce epilepsy and you produce a form of temporal lobe epilepsy in a rodent. These are both well-established models of epilepsy that are widely used to discover new therapies that go into the clinic. And what's most important to point out in the details of these papers is that when we transplant inhibitory interneurons derived from the medial ganglionic eminence, or MGE, we can dramatically reduce their epilepsy. So in a genetic epilepsy mouse, looking at EEG events to document the number of spontaneous seizures, we found a 96% reduction in seizures. In the acquired models, we found 88 and 92% reductions up to 6 months after we transplanted, which is pretty long in the life of an experimental animal. And interestingly, especially in the acquired epilepsy models where you see what are called comorbidities, similar to patients, other problems the animals have that you can measure both behaviorally or using cognitive tests, all of those deficits were also rescued by our MG transplants. And so the success of this project, of course, is at a preclinical level, and it involved at this point taking rodent tissue and transplanting it to other rodents. But how do you get this to a large animal? And more importantly, how do you eventually get this into people? So one of the issues is that you'd want to be able to test this in a larger animal that's having a form of epilepsy that resembles temporal lobe epilepsy. Interestingly, those already exist here in California. So with global warming and other man-made issues, there's an increase in the temperature of the Pacific Ocean. This leads every summer to harmful algae blooms. The algae concentrates something called domoic acid, which is a neurotoxin that gets in the food chain and eventually gets all the way up to sea lions and sea otters. The animals that ingest these toxins through the algae blooms eventually develop hippocampal sclerosis, interneuron loss, and adult onset epilepsy. These animals have also been seen to show high mortality, difficulties in navigation, anorexia, atypical behaviors, and cognitive impairments. So they're basically a naturally occurring wildlife animal that has epilepsy that resembles a human form of epilepsy. And we know this through decades of work by Paul Buckmaster and others, looking at the brains of animals that were deceased or died from this domoic acid poisoning and epilepsy. And so we've seen consistently that the hippocampus is smaller, it atrophies just like in humans. You can see that on the right side compared to the left side. And more importantly, that there's many features of this brain that resemble what we see in humans with temporal lobe epilepsy. Again, this is beautiful work, seminal work by Paul Buckmaster, a colleague of mine down at Stanford University here in California, who studied these animals with the help of the marine mammal centers here in California for almost 2 decades now. And what Paul has found is that there was a loss of neurons that specifically matches the types of loss we see in humans with TLE, that there's synaptic reorganization, what's called mossy fiber sprouting, that also resembles what we see in humans with temporal lobe epilepsy. And that specifically, it's unilateral like patients, and it's a specific loss of the two types of interneurons that we can make with MG cells, parvalbumin and somatostatin. So this became, in my kind of view, a couple of years ago, an ideal opportunity to potentially develop this treatment and bring it to a larger species. And so we started to work with the marine mammal centers and other veterinarians in the area. And that came over a couple of years of progress to a point last summer where we were introduced to Cronutt. Cronutt is a sea lion. He's about 7 years old, living here in California. He was rescued by the marine mammal centers on 3 separate occasions, each time because he beached himself and was having convulsive seizures that people could see. The rescue centers tried to treat him with anticonvulsant drugs and eventually realized they could not release him back into the wild. He was then adopted by Six Flags Discovery Park here in Vallejo, California, which has a sea lion park. And because of that, he was able to get MRIs on his brain through a study with imaging colleagues at University of California, Berkeley, including Ben Inglis. And so what they were monitoring was the damage to his hippocampus. Based on Paul's work and other work over the years, we knew that domoic acid would cause atrophy or shrinking of his hippocampus. And so they monitored that for several years. And of course, they watched his behavior, which increasingly got worse. So initially, they only would see 1 seizure occasionally. In the first year, 2018, by 2019, he was having several seizures a year. These were not treatable by antiepileptic drugs. He takes both phenobarbital and diazepam. And as it progressed, last September, his seizures were so severe that he was losing weight and anorexic. In fact, when I first met Cronutt in late September of last year, he hadn't eaten in about 25 days. And so his handlers had deemed he was in a life-threatening condition, and his only option was either a novel therapy, one-time therapy like ours, or they would have to sacrifice him, unfortunately. And so how do we get to our rodent experimental studies that we do in laboratories to a large wildlife animal that we've never really worked with? Well, one of the things we needed to do was adapt a stereotactic technique. So the way this process of interneuron transplantation works, of course, is we deposit a very small number of these MG cells into a very specific region in the hippocampus, in the brain of the host animals, and then we let the cells migrate. So we had to initially come up with an approach to adapt this to a large animal. This was not trivial and didn't exist when we had started this, but we've reasoned based on how neurosurgical techniques are done here and in other places around the world, that we could use a stereotactic technique if we had the right coordinates to know where in the brain and what landmarks we could use to target the correct hippocampus. To do that, we initially made measurements of skulls of sea lions that are stored here at the Cal Academy down the street from UCSF. And we worked with imaging experts to look at MRIs and CT scans that would allow us to guide this procedure on site. And so this is what some of that preliminary work looked like. Of course, it involved, knowing which hippocampus was damaged. This is actually a MRI scan from 2018 on Cronutt. And you can see on the far right side, this little area where it's a little bit whiter. This is where you have hippocampal atrophy. And that gave us a target for where we would deposit cells. Simultaneous to that, of course, we had to develop a cell source. Where we would find MG cells, fetal cells that we could transplant in these animals. Turns out we can get them from pigs. Pig donors are not unusual. In fact, early heart valve transplants came from pigs. And so we reasoned that it would make sense to use fetal pig tissue to harvest MG cells. Mariana Casalia, a very talented postdoc in my laboratory, spent 4 or 5 years tracking down where we can get pig embryos, both with the veterinary clinics at UC Davis nearby and the National Marine Swine Center here in America. And then finding out whether these cells mimicked the other types of MG cells we had done with rodents. Long story short, fetal pig-derived MG cells look just like rodent MG-derived cells. And so that allowed us to have a cell source that we could transplant into Cronutt combined with this stereotactic technique. So as some of you might have seen, Cronutt was operated on back in October. It was a one-day procedure. It was a heroic effort by everyone involved. There were about 20 or so people who came together during COVID at a time when we were limited in how many people we can have in the room for these procedures. His handlers drove him down from Vallejo to a veterinary clinic in Redwood City, about 40 miles from where he lives. He was intubated and prepped, anesthetized and prepped for surgery. He had a CT scan done on site. And once everything was in place, Mariana and the neurosurgeon residents, Ryan Kachansky and John Andrews, with Mariana's help, transplanted pig MGE cells, progenitor cells. These are not stem cells. This is actual fetal tissue from a pig into the brain of Cronutt. His pictures were taken. His article appeared in the New York Times telling his full story. And he went back home where he's been recovering nicely. So this data now is a little bit older, but this is from 6 months. He's now at 8 months. And you can see this is his weight loss, which was precipitous in the time before the transplant, which is indicated here at time 0. In fact, he had lost 25% of his weight before the procedure. Since then, he not only has gained his weight back. Last time I talked to the handlers at Six Flags, they said he was, quote, fatter than ever. And so he's doing very well on weight gain. He has had no abnormal neurological signs or behaviors. And most importantly, for the success of this N of 1 compassionate use trial on Cronutt, he is now about 8 months completely seizure-free following his interneuron transplantation therapy. And so I just want to thank, of course, the many, many people. This is only a fraction of those who came together to make this happen. Most notably, Claire Simeon and her husband, Sean, who are the vets who were his primary handlers, and the great crew at Six Flags, neurosurgeons, Mariana Casalia, who did heroic work to make the pig cell project work. And of course, Cronutt himself, who has been a willing and happy participant and is thriving since his procedure. And I think that's it for my slides.
Thank you so much, Scott. That's absolutely amazing. I really am so impressed that you've had such success, and well done, Cronutt, for being seizure-free for so long. Such good news. So if you can hang on for a few minutes, we have some questions coming in. And so some from staff here and some from the audience. So first, and possibly the most one on all of our minds, is given the success that you've had with Cronutt, do you plan to roll this out with more sea lions?
Yeah, that's a great question. So of course, sea lions are not experimental animals. They're wildlife. So we have to be careful on which ones and who we could work with. The ones that are rescued by the rehabilitation centers are released back into the wild. So we have no plans to transplant animals that will be released back into the wild. Mm-hmm. That said, hundreds of sea lions are domoic acid poisoned every year. And that's only increasing, like I said, because of climate change. And we're talking to some of the centers that have long-term care animals in forever homes that are domoic acid poisoned. And so we're waiting, of course, to see a little bit longer how Cronutt does, but certainly there's a potential to do additional sea lions that are in captivity for other reasons. Fantastic.
Good luck with that.
Thanks.
And then the next step, of course, which I think is very much on everyone's mind, is, then we hope that this will translate to success in treating people. How soon do you think it could reach the clinic in humans?
Yeah, that's a great question. Of course, our ultimate goal is twofold. We of course want to help the sea lions and other wildlife that are having epilepsy here on the West Coast. And of course, we'd eventually like to see this translated to patients. I'm always cautious. It's hard to tell timeframes. It's hard to give timeframes. Sometimes science leaps ahead and sometimes it goes a little bit slower. So I'd be hesitant to put a year on that. I think we're moving in the right directions with this project. I think there's certainly precedence for using fetal cells. They've been used in Parkinson's disease successfully in the past, and there's certainly a precedent for using pig donors. And so I think as those two things kind of coalesce and we get more proof of principle data and safety data, of course, which is a critical other feature that we'd want to see over time with the sea lions, that eventually this could progress to the clinic, theoretically.
Great, great. So we have a couple of questions from the audience. One from Caroline is, might this work for epilepsy, which occurs in other parts of the brain?
Certainly. So there's no, the fascinating thing about MGE cell transplants is where you can put them anywhere and the cells will migrate. They have an endogenous ability to migrate. In rodent models, we put them in very different regions of the brain and the spinal cord, and for different types of indications, and they work very well. So as long as there's a specific focus that we can target that we think is generating the seizures, it would be amenable to a MG transplant.
Yeah, that's really exciting. So another question from the audience about Cronutt from Tara is Cronutt still on anti-epileptic drugs, or has he been able to cut back?
Right, such a great question. And we go back and forth with the neurologists on what people with temporal lobectomies who get resections do. As some of you know, as clinicians especially, there's a very mixed data on how their AED regimen goes. Many times they stay on their AEDs as long as there's no problems. And so that's our initial goal with Cronutt, to remain on his low doses of AEDs and not change anything. As we watch that literature in humans and we kind of discuss this with more clinicians, we could readjust that. But at the moment, the consensus among the neurologists we spoke to who are experts on that, on TLE, is that patients who have resections stay on their AEDs for 1 or 2 years at least. And many times they only come off AEDs because they want to drive a car. And fortunately, Cronutt will not be asking us to drive. So I think it's possible, as long as he's doing well, not to change too many things.
But he can swim.
He swim. Yes, he's perfectly happy. He lives in his apartment in Vallejo, which has a pool and a dry dock area as well.
Super. So a question from Aswin is, and this is a really interesting perspective. Will it be possible, or is it possible, to induce human-derived stem cells into MGE type interneurons?
In my opinion, no. There's been tremendous amount of work and hype around that for about 2 decades, parallel to our work. From what I've seen in the literature and the different people working on that and the different companies working on that, no one has been able to reproduce successfully making MGE cells that have both the migratory properties that fetal tissue has, the integrative properties, and most importantly, form the correct types of interneurons. So getting parvalbumin types of interneurons, which is a detail, has never been successfully shown for human-induced cells. And so those critical features make it hard to see how that's going to change with the present technologies. I don't want to say in the future that might not, there could be a breakthrough. The other very critical difference, and I really can't underemphasize this, is with these fetal embryonic tissue transplants that we do, we have never seen a tumor in hundreds and hundreds of preclinical cases. But induced pluripotent stem cells, especially those derived from humans that are transplanted, there's certainly a risk of tumor formation in almost every type of protocol you use. Occasionally, you'll see an animal with tumors. And so until that part is also solved and you get the correct types of interneurons, I would say I'm pessimistic, I guess, about the current technologies to derive MG cells from human interneurons, from human stem cells.
Yeah, and I'll just echo, my area of research is quite different, but we also have real concerns about getting the exact right types of interneurons, because there's so many different types of interneurons, and the stem cells, you can make them into neurons, but you're never quite sure they're exactly who you want them to be. So I appreciate that.
Thanks. And I'll add when we've made other types of interneurons using different colloidal ganglionic eminence, when we've made the type that resemble the human versions that you get. And we put those in wild type animals. Those animals develop seizures. And so you have to be very careful. If you make the wrong types of interneurons, they're going to inhibit other inhibitory cells. And that will create a condition that'll be worse.
It'll be the absolute opposite effect of what you wanted. So it really is important to get those cells. Yeah. So just another question that's coming up is, so we do understand that Cronutt has a Twitter profile. So are you behind his Twitter account?
No. So I run my lab account, Baraban Lab Twitter account. I do not run the Cronutt Twitter account. And in conversations with my lab members, my family, my friends, the people at Six Flags, the other vets involved, we've been unable to figure out who, or at least no one's admitted that they're running the account. But it's not me and it's not my lab. That much I'm sure of. So yes, I would love to know who's behind the Twitter account. If they're watching this today, maybe they'll drop a hint later on. But it's certainly not us. So
perhaps we need to start a Who is Cronutt campaign.
Yes.
This mystery tweeter, the real Cronutt tweet. And then I just want to finish with one final question, which we ask everyone who gives a webinar, just a chance for you to take a step back and talk about the big picture for our audience. And the question is what are your hopes for the future of research in epilepsy?
Mm-hmm. So I usually look at that kind of twofold. I mean, the immediate hopes for which I kind of have some control over to some extent is what my lab will work on. I strive to do a couple things here, but mostly we try to push the envelope on translational approaches that will someday help people with epilepsy. So as you know, we work on these MG transplants, which we think are a potential one-time treatment for acquired forms of epilepsy primarily. And we also work on zebrafish models representing genetic forms of epilepsy that kids have. And we've used those for drug discovery. And so my immediate goals for the lab is to obviously continue in those directions and expand them as much as possible through our work and collaborations that we have around the world and kind of push that forward. My bigger hope as a member of the epilepsy community, which dates back now almost 30 years to when I was a graduate student, is that these types of research programs that we have and we publish on and that I talk about publicly inspire a younger generation to take more risks and be more innovative in the epilepsy community. My overall, frustration, if I could call it that, in the epilepsy world is that a lot of these types of projects do not change the needle, don't influence too many other labs to jump in and do something similar or something even more innovative than we do. And so my hope for the future of epilepsy research is that the young people, the young investigators, the kids who are in graduate schools and postdocs who see these types of talks and read our papers, that they're influenced to take these risks, because that's how we got here. That's how I got here. I got here by taking risks on projects that I thought had great potential to move the field and then pursuing them rigorously with scientific rigor and careful kind of steps. And that's what leads us to things like Cronutt, which looks like it was a one-time procedure that we just magically did, but really it was 15 years of work. And so I hope that young scientists in the epilepsy community can see that and be inspired to take those types of risks in their own career. Because I think moving the field forward as a whole is the other hope I always have as a professor.
Yeah, and just to emphasize for members of the audience who aren't aware that you actually are exceptional in that you're breaking boundaries in two completely different areas, both the Cronutt story, which as you point out, started long before there was any option of treating a sea lion on the horizon, and also in your really groundbreaking work on looking at epilepsy in the zebrafish model, which has given already massive new insights, including to really devastating childhood epilepsies, which we would not have had if you hadn't gone and broken the mold and used a different model.
Thank you. Appreciate that kind word about it work.
No, it's been fantastic. And thank you so much for joining us and telling us about Cronutt. And we do hope that Cronutt continues to be seizure-free and swimming and putting on weight, as much weight as a sea lion should put on, which I understand is quite a lot. and that your research will translate, as fast as research is safe to do to people with epilepsy. And it is looking incredibly promising. So all fingers crossed there. And thank you also to all of our viewers for tuning in. We hope that you will also join us. And thank you also, viewers, for all the excellent questions. Really, really helpful and thoughtful. And we hope you'll also join us tomorrow, Wednesday, 2 p.m. UK time, for our Hot Topics webinar on the facts of cannabinoids and epilepsy. So that's a really interesting one because there's an awful lot of non-facts out there. So this will be a chance to get the real facts as opposed to the alternative facts about cannabinoids with epilepsy from people who've really been leading in the field. And then on Thursday at 2 p.m., also UK time, for our Research Roundtable webinar, which is a really exciting opportunity to hear from all of this year's 2021 research awardees from Epilepsy Research UK, which is people doing a whole range of research into new treatments for epilepsy, new ways of diagnosing patients, trying to harness some of the video technology we've developed during COVID to actually help patients get diagnosed without having to come into the clinic. So some real exciting things happening. And the links to register for both of these webinars are going to be posted in the chat. And I hope you all will jump in. And I see they are already posted. And I hope you all jump in. And thank you once more, Scott, for really an exciting and inspiring story. And thank you everyone for joining us, and enjoy the rest of your afternoon or day, depending on where you are. Thank you.
Thanks for having me. Take care.
Yep, you too.